Uh, Dr. Roozbeh is an applied scientist at a company called Light Matter in Boston. He's also the co-founder of Extrapol AI. The work that he's presenting today was done when he was a postdoc at Yale University at the VA healthcare system. Before doing a postdoc at Yale, he did his PhD in computer science at the University of Maryland. His research interests are scientific computing, deep learning, and machine learning. He is going to talk about community detection in medical image datasets using wavelets and spectral methods.
Hi, everyone. Hi, everyone. Thank you so much for the introduction. It's great to be here. As my talk is about community detection in medical image datasets. So here I'm dealing with two research questions. The first research question of this work is heterogeneity in image datasets. So when we deal with the image datasets, we usually like to group them, label them. This is usually motivated by from the clinical perspective, not the machine learning. For example, the clinical procedures that should follow are different. We have different types of tissue we like to group the images into. But here in this plot, I'm showing all images that are labeled at the same type of tissue in the context of colorectal cancer. So when we label these images, sometimes when we go to the domain of machine learning, a lot of information is lost because like all these images, they have some intricate details, and from the eye of the model, all of these are the same thing. So it's good to have computational methods that can distinguish these differences, identify these differences, without us telling them exactly. So this is the first research question. The second research question is inferring the disease spectrum. So as I mentioned, we might label images. For example, these are CT scan images. We label them as patients with COVID or non-COVID. But not everybody who has COVID have it with the same severity. So it's also useful to be able to automatically put the images on a disease spectrum. So this is not providing insights exactly to medical experts. Medical experts look at these images. They can immediately say which one is the severe one, which one is not. But we are not always dealing with one or two images. We are dealing with sometimes thousands of images, a very large number of images that are not labeled, and that medical experts, their time is very valuable. So having a medical expert going through a few thousand images one by one, it's very expensive, time consuming, and sometimes even prone to mistakes. So it's good to have procedures, computational procedures that can automatically process images, analyze them, distinguish these differences, and maybe put them as the starting point for the medical expert to go over these images once they are labeled. Once they're grouped and organized, and then maybe the medical expert can come and might have spend much less time processing these. So the method I'm proposing here uses wavelets to analyze the images. Wavelets are a class of functions for image processing and feature extraction. They're also used for signal processing. They have a solid foundation in mathematics. They have been around since early 1990s. And they have a similar computational nature as convolutional neural networks. So nowadays we're using deep learning quite a lot. We use trained models to extract features from images. But not always we have an appropriate convolutional neural network that is appropriate for to be used in a specific medical context. So if you are at a medical institution, you might pull all images of certain type for patients of certain disease, And it's not always, it's not like you have always access to a convolutional network that is trained and appropriate to be applied readily for this application. If you already had some model that could classify the images, maybe that's perfect, but often we don't have that. And those models are usually very expensive to come by and also very computationally expensive. They need specific computer infrastructure for you to be able to use it. But wavelets are quite inexpensive computation. So the way it works is that you decompose an image, you get a set of wavelet coefficients. This operation is reversible. So from the wavelet coefficients, you can reconstruct the image, but you can also consider those wavelet coefficients as features from the images. Then you can put those wavelet coefficients into a matrix or a tensor, then you can analyze the patterns in them. Maybe there are some redundancies, you can discard them. And then at the end, you have a set of wavelet coefficients for each image. This is exactly what we do. And then once we have those wavelet coefficients, we compare the images based on that, their similarities. We come up with a distance matrix, then we convolve that distance matrix with a Gaussian kernel to infer a similarity matrix. And then we, from that, we compute a graph Laplacian and analyze its eigenvalues to estimate the number of communities in the image data set. And we proceed with the spectral decomposition and spectral clustering, and we cluster the images. So this is the formal algorithm we have. The code is also available on GitHub. So I'm going to move on to present some results. So this is the first data set. This is a data set for COVID patients. It's about 900 images. It's a mixture of different types of images for patients with COVID or not. And also we have information as to whether the patients have developed pneumonia or not. So we throw all these images to the algorithm without telling the algorithm anything about the patient. We just give the images to it. The algorithm processes the images, compares them, comes up with this similarity matrix. Then we look at the eigenvalues of the graph Laplacian. We choose using a threshold on the eigenvalues. We choose 25 clusters. It's quite on the conservative side where we can choose even less than that. And then we cluster those images based on that. So this is the same similarity matrix I showed earlier, this one. It's just reordered based on the similarity of images. So on the diagonals, each box, you see each group of similar images. And on the off diagonal, you see the similarity of each of the clusters to each other. When we look inside these clusters, we see that actually images of similar kind have appeared inside each of those clusters. And then when we look at the biomarkers, we see that only the images who were for patients who had developed pneumonia only appear in 6 of these clusters out of the 25. So so this clustering has actually picked up those biomarkers that has to do with pneumonia. The second data set has to do with colorectal cancer. I already showed this hint of it earlier. There are 9 types of tissue. For some of the tissues, we don't see much varieties there. Still, the algorithm can group the images that this one at the bottom where we see the blood vessels. But for some other types, this is a cancerous tissue. We see there are a lot of varieties that the algorithm can pick, and these are all the same and one kind, but we can see these differences that the algorithm can pick and group them. This could be related to different severities or aggressiveness of these. This is something we intend to investigate further for this for this specific study. But we can also take this one step further and analyze the cross similarities. Like one class analyze the similarity of images, not just within the class, but the similarity of images to the other class. So this is the third data set. It's about COVID and non-COVID, only CT scan images. And then when we perform this similarity analysis, an image within one group, for example, the COVID class, that has strong ties to the images in the COVID class, but weak ties to the images in the non-COVID class. Such images would go to the far end of the spectrum on the right, and then the opposite on the left. And the images that have strong ties to images to the other class, they go to the borderline. And this is also helpful to identify images that are at the borderline, because these are the images that the model could make a mistake when classifying them. These are the images that maybe are early signs of COVID, that are ambiguous. Maybe a doctor wants to take a second look and such. So just to summarize what I explained, we have this algorithm that can identify a group of images. For unlabeled images, we cluster them. It's expensive to label them by medical experts, so we can use this algorithm to group the images as a starting point so that the medical expert can go over them more later on, not just go over the groups of images. We can also use this for cross-class similarities. For labeled images, we can use it to identify infer disease spectrum. There are also applications such as triplet mining, which require identifying images of similar within the similar group as an anchor image, and images from the other groups for training a model with triplet loss. So this is also a future direction of research. Thank you very much. I think I'm at the 10 minutes. I'd be happy to answer any questions.
Thank you very much. Is there any questions? If there is no questions, we can move to the second speaker, Dr. Zendian Deng. Just a minute.